Hey class, welcome to Chapter 9. We're going to talk a bit about transcriptional control of gene expression. So these are the factors that are correlated with the amount of protein abundance within a cell. So the number one thing that results in increased or decreased protein abundance is the rate of gene transcription. So the more genes that are transcribed for a specific or the more transcripts that are made for a specific gene that result in this protein means that the higher the amount of those transcripts, the higher the amount of the protein. The lower the amount of transcripts, the lower amount of protein. But there are other influences as well that can change how much protein is in a cell. One is once that mRNA is made, is transcribed, how much of that mRNA is actually translated. So changes in the amount of translation of those transcripts can lead to increases or decreases in protein. The amount of protein that's degrading. So if proteins are being broken down at a faster rate than they are being made, then that changes the abundance. And then lastly, the mRNA degradation. So how quickly those mRNAs are being broken down, either before they get to the translation phase, or if they're hanging around extra after and are translated multiple times, which usually doesn't happen. But these all play roles in how much protein abundance is in a cell. So mutations and transcription factors can lead to changes in gene expression for multiple genes because these transcription factors are involved with or the actual machinery that lead to transcription of genes. So improper regulation of these mechanisms controlling this transcription can lead to changes in phenotype. So here we have an example of the human HOXD13 dominant mutation. Let me indicate it right there. And this leads to polydactyly in humans where, in this case, an infant has an extra digit on the foot. We also have an example here with the Ubx gene, which is a homozygous recessive mutation in Drosophila. This Ubx gene prevents expression of thoracic segments in Drosophila. So normally these Drosophila have this organ called a haltere, which is a balancing organ. But in the case of the Ubx mutation, that haltere is turned into a copy of the thoracic segment in front of it. So this segment is duplicated to right there, and we end up having a Drosophila that has an extra set of wings. And then lastly, we have inactivation of a copy of, well, a couple different genes here, AP2-1, PI1, and AG1 genes here in Arabidopsis, the plant that I work with. And what this does is it prevents the flower from creating these floral identity genes being expressed. So instead, the flower or the tissue that's supposed to develop into the flower develops into these leaf-like structures. So let's look at an overview of eukaryotic transcription control. And so the condensation or condensing of chromatin prevents things like Rna Polymerase and general transcription factors from being able to access a gene, and therefore that gene can't be expressed and turned into a protein. And so repressor proteins have a role in this. And so what repressor proteins do is they can either bind to transcription control elements to inhibit transcription initiation by this POL2 enzyme that we'll get into shortly, or they can interact with this multi-protein co-repressor complex that leads to more condensation of the chromatin. And so, but once genes are going to be turned on, what happens is these pioneer transcription factors come in, and they will bind to the specific regulatory sequence within this condensed chromosome or chromatin, excuse me. As you can see, they're kind of buried underneath here. And they interact with these chromatin remodeling enzymes, or chromatin co-activators here, as well as acetylases that will add these acetyl groups to those histone tails and allow that chromatin to kind of slide and allow access for these transcription factors and polymerase to come in. And so once that occurs, we have activator proteins right here. And the activator proteins bind to a specific transcription control element in both the proximal promoter sites, so real close to the promoter, as well as very distant sites. So you can see here they are kind of bound to something right by where the gene is going to be transcribed, as well as areas that are distant. They interact with one another and with that multi-subunit mediator complex, which is this whole big globular thing here. And they work to assemble general transcription factors and RNA polymerase into the correct position. And so once that occurs, we have activation of transcription, where that POL2 enzyme comes in and it starts the process of transcribing a gene. But after about roughly 100 nucleotides, give or take, there is another enzyme that comes in called negative elongation factor, or NELF. Where is NELF? Right here. And what NELF does, it associates with DSIF, which is DRB sensitivity inducing factor, which is right below it there, and they cause transcription to pause. And so once that occurs, this POL2 NELF DSIF complex associates with PTEFB, which is a cyclin TCDK9 kinase. But what it does is it releases that NELF, allowing transcription to resume. So then we go back up here and we're transcribing again. So it's kind of a fail-safe or a pausing in transcription to make sure that the gene that we want to transcribe is being transcribed and it didn't happen erroneously. Okay, so very briefly, I'm going to hit on the Lac Operon here. Hopefully you've covered it in your genetics class. If not, in my genetics lectures on my YouTube channel, go to the regulation of transcription, and the Lac Operon is in there, and it goes into much greater detail, but we're going to breeze through it here real quick. So the Lac Operon has a cap site, which is bound by enhancers. It has a promoter where the transcription factors and the RNA polymerase bind. It has an operator, which is where repressors bind, and then it has an operon. So it's got a suite of genes that are all required for metabolism of lactose. Now, this setup allows for the actual sensing of the environment and knowing when to use genes specifically based on if that lactose is present or not. So in our first example here, we have no lactose and we have high glucose. Now a cell like E. coli preferentially wants to metabolize glucose because it's the most energetically favorable and you get the most AMP, or the most calories out of metabolizing glucose compared to lactose. So if that's the case, then we have this repressor that comes in and it will bind to the operator and it will prevent those lactose digesting genes or metabolizing genes from being expressed, because we don't have lactose. Why make those proteins, right? And so in the second case here, we have lactose and glucose present. Now, like I said, we preferentially want to digest or metabolize glucose. So if we have lactose present, what happens is the repressor does not bind, because we need some of those lac genes to get rid of the lactose, but the activator, this cap, which is associated with cyclic AMP, that doesn't bind either. So what we get is this kind of medium to low level of transcription of the lactose operon. And the reason for that is we need to kind of start getting rid of a little of that lactose because we don't want it to build up and it's in the environment. But we don't want to go overboard and make a ton of genes for lactose metabolism because it's not as energetically favorable as glucose. So it's more of like a management approach. And then lastly, what happens if all we have is lactose and no glucose? So we're desperate to metabolize these sugars for energy. So what happens then is, because there's no glucose, this cyclic AMP is bound to the cap protein, which is an activator, so it actually increases the amount of transcription of a gene. In addition, there is no binding of the repressor because there is lactose, so that removes the repressor, and that allows for a very high amount of transcription of this Lac Operon to produce or to process all that lactose. So prokaryotes, like E. coli, they only have one polymerase for the purpose of making our mRNA. And so there's got to be something, though, that kind of specifies where the polymerase goes, right? Since there's only one of them, it needs to know what genes to turn on, what genes to turn off. And so in addition to the regulation of expression that we saw in the last slide with the repressors and activators, there's also something called these sigma factors. And what these sigma factors do is they are there to turn on, they're kind of like the on-off switch for this polymerase. And they are associated with certain groups of genes. And so when they bind to that polymerase, or when they get turned or increased expression for these sigma factors, those specific genes associated with that sigma factor will be expressed. So take a step back. Let's look at this as an example here. So we have sigma factor 70. And so this sigma factor will interact with polymerase to turn on housekeeping genes and most genes that are involved in cells that are replicating. And so whenever that sigma 70 binds to polymerase, it is making genes for those purposes. And so in addition, we have sigma S, which is, instead of replicating, it's the stationary phase, so cells that are just kind of sitting there and doing their day-to-day but not replicating and whatnot. That S sigma factor will make genes like general stress response and general maintenance of the cell. We'll skip down here. How about sigma F? So this is genes that are involved in flagellum assembly. So if you're a cell that has a flagella that you're using it to kind of get around, you don't want those genes turned on all the time, right? Because you only need to make a flagellum once. You might have to repair it at some point, but you really only need to make it once at the beginning of your life cycle. And so why you don't want them turned on all the time. And so the sigma factor F is used probably very early in development to develop that flagellum, and then generally is likely not used anymore. So this adds a little more specificity to the expression of certain genes within prokaryotes. Okay, we're going to talk here about a two-component system that regulates gene responses. And in this example here, we're going to use glutamine. So when there are high amounts of glutamine in the cytoplasm of a cell, we have this NTRB complex. And NTRB has a sensory domain, and this sensory domain binds that glutamine. And so if there's a lot of glutamine, a lot of those sensory domains will all be filled, and we get this kind of horseshoe conformation here that is the inactive state of this NTRB. And so we also have this NTRC, which has a regulatory domain and a DNA binding domain. And in its high glutamine state, they are kind of also in a horseshoe state where they're bound together and inactive. And so when glutamine is low, what happens is the glutamine is either not present or released from the sensory domain, which allows it to form this, change its conformation, and in this example form kind of this more linear protein complex. And that causes this phosphorylation through a protein kinase transmitter domain that transfers this phosphate from to a conserved histidine. And this conserved histidine is this H here. And so once that happens, the phosphate then is transferred to this aspartic acid or D domain in the regulatory domain of the NTRC. And that converts the NTRC into this active form that we see over here. So it linearizes it. And when that occurs, that frees up that DNA binding domain to attach to the enhancer site upstream of the promoter and then bring those transcription factors and the DNA, kind of everything together, right? So enhancers help to bring together all the required components of transcription. And so on the bottom here, we just have kind of a generalized version of this where we show that we have, a sensory domain, this histidine kinase sensor, and then we have this sensor domain that's unbound, etc. So it's just a generalized version of what we just covered. Okay, so let's talk a little bit here about ribose switches. And so ribose switches control transcription termination. And so they attenuate transcription elongation of some operons and single gene encoding enzymes that are involved in biosynthesis of amino acids and other metabolites. And they're sequences of RNA that are most commonly found on the 5' UTR, so upstream, untranslated region of bacterial mRNAs. And they fold into these complex tertiary structures that are called aptamers. So we see here this folding of an aptamer. And these bind small molecule metabolites when these metabolites are present in high quantities. And so looking at this process here, we're looking at the Bacillus subtilis is the prokaryote, and this is the XP because I don't think this is written on here, XPT PBUX. That's a dash in between. This isn't even worth me doing, it looks so bad. PBUX operon, which encodes enzymes that are involved in purine synthesis. So that's the most important part. And so we have this 5' UTR that has a ribose switch, and this can fold into alternative structures depending on the concentration of those purines. And so we have this confirmation right here that if there's a low purine concentration, and in this high purine concentration, you see that a purine is actually bound to where this kind of fork is in the RNA. And so this stem loop transcription termination signal is created where we have a stem loop followed by a bunch of U's. That is created only when this purine is able to bind to that aptamer. And so that's a signal that they should not create more or transcribe more. And so when the purine concentration is low, there's an alternative RNA structure, which we have right up here at the top. And this prevents formation of that transcription termination signal, that stem loop followed by a bunch of U's. And that leads to increased transcription of the operon. And so as you can see here, the high concentrations of purine down at the bottom, that blue followed, the blue stem loop there followed by the U's is the signal to stop transcription, while that same structure is not present in the low concentrations of purine, which allows continued transcription of the gene. Now this here, part B, this is just a crystallized structure showing that RNA bound with those purines in the center. So this is likely review, but this is a good informative slide here that we can quickly hit on. So there are three classes of RNA polymerases that are transcribed by eukaryotes. RNA polymerase 1, 2, and 3, and they all have different or slightly different functions. So RNA polymerase 1, it helps to make pre-RNA for what will eventually become rRNAs. So they make it's responsible for transcribing the genes for the ribosomal components and protein synthesis. RNA polymerase 2 is the one that we talk about a lot, that is responsible for making mRNA, which encodes proteins, but also a lot of these non-coding RNAs that have functions, or functional RNA as we call them, which are involved in RNA splicing, chromatin-mediated repression, and translation control. And then lastly, we have RNA polymerase 3, which is involved with the tRNA, so those RNA that go and find those amino acids to bring them to the proteins for protein transcription. So they're involved with protein transcription, but also RNA splicing and other small stable RNAs that have various functions and a lot of unknown functions at this point. Okay, so if we were to look at the polymerases and compare them between eukaryotes and prokaryotes, here we have an example of bacterial RNA polymerase, and then remember there's only one in bacteria, and then we have yeast RNA polymerase 2. You can see that the structure is very much conserved between the two. So polymerases are a very highly conserved protein complex throughout all of life, for that matter. And so, as you can see, as we get, quote, higher up the evolutionary ladder, there becomes more complexity to the yeast RNA polymerase compared to that bacterial RNA polymerase. However, the overall structure, as you can see, is greatly conserved. Okay, so let's look at the actual structure of polymerase. So let's get into the weeds a little bit here. And so on the first left-hand side here, we have free RNA polymerase. And as you can see, we're going to focus to start here on this clamp domain. And when it's not associated with DNA, this clamp domain is kind of in this open position. And then when the RNA polymerase interacts with DNA, the clamp domain swings over the left cleft and will kind of chomp down, so to speak, on the DNA. And this happens during transcription, when this polymerase is doing its job. And it will trap that template DNA strand and the transcript as it's being made within the polymerase. And so binding of this clamp domain to 8 or 9 base pairs of this DNA RNA hybrid, so where is it actually being transcribed, may help couple the clamp closing to the presence of RNA. So it requires that RNA is starting to be made for this clamp to remain closed. As you can also see, we have this magnesium ion in the middle here. It's kind of covered up in the transcribing RNA image on the right hand side, but this participates in the catalyzing of those phosphodiester bonds that are required for transcription. We also have this RPB2 domain, which is right here, and this forces the template DNA strand entering the jaws of this polymerase to bend before it exits. So it's kind of a conformational subunit that helps to orient DNA properly for the transcription. And then we have this bridge alpha helices, which is this green unit in the center here. And this extends across that cleft, or the gap of the polymerase. And it's thought or postulated to bend and straighten as this polymerase translocates one base down the template strand. So it's kind of like a ratchet, supposedly. It's theorized. It's not 100% proven. And then the non-template strand of the DNA that is not being transcribed, it forms this flexible single-stranded region above the cleft, and it's not shown here, but it extends from 3 bases downstream of the template base paired to the 3' groin end. So it's just not really involved, I guess is the best way to put it. But then it comes back together as the polymerase transcribes that gene. And so it will exit the same way that the rest of the transcribed gene does. Okay, so let's look at promoter elements excluding CpG islands for metazoans or animals. And so RNA polymerase II needs these promoter conserved sequences to be able to bind to start transcription. So these have to be present in all genes that are going to be turned into mRNA and then into a protein. So there are several different types of gene sequences. We have TATA boxes, we have initiator sequences, we have downstream promoter elements, as well as TF2B recognition elements. So this is a site where the generalized transcription factor TF2B recognizes and binds to. And so the TATA box here, you'll notice that there are these letters, and this is the sequence that these promoter elements make up. So you see that there's the TATA, obviously where TATA box gets its name. And then from there, we have a couple of different options. The 5th base can be an A or a T. The 6th has to be an A. And then we have a couple other options further down the line. And these are very, very highly conserved across all animals. This RNA polymerase needs that recognition site to know it's in the right spot to start transcribing a gene. And if you change those from these conserved sequences that we see here, we start to get decreased transcription because the RNA is not going to bind as sufficiently. Now the initiator sequences, as you can see, there's a lot more variation between the different organisms. It's not as highly conserved as the TATA boxes. So let's talk a little bit about chromatin immunoparticipation technique, or CHIP assays. And these are assays that localize where a protein of interest associates within the genome, so specifically DNA binding proteins. And so for this one, or this example here, we're doing chromatin immunoprecipitation using an antibody to RNA polymerase II that gives us evidence for where CpG islands come into play in metazoan or mammals. And so in this experiment, for step 1, let me get my marker, live cells are treated with 1% formaldehyde, which covalently cross-links proteins to the DNA and proteins to proteins, so it kind of freezes everything a protein is touching kind of in place. And then the preparation, or the cells are lysed or sonicated, and solubilized chromatin is used and shears this DNA into 200 to 500 base pair fragments lengthwise, so relatively small fragments of DNA. And then in step 3, I should be marking these, shouldn't I? In step 3, the anti-RNA polymerase II antibody is applied and precipitates RNA polymerase II and its covalently linked DNA. So we're going to get rid of all the transcription factors and other proteins that are interacting with the DNA, the histones, etc., and only pull down, using these antibodies, the RNA polymerase II. And then what we do is we reverse the cross-linking, so you can use a base or something of that nature to get rid of that cross-linking that we did with the formaldehyde, and then we only have the DNA that polymerase was binding to. And so from there, we can look at the DNA regions that polymerase recognize. And so in this example down at the bottom here, they did RNA-seq or DNA sequencing and looked at where polymerase was binding in mice. So let's talk about how this RNA polymerase II pre-initiation complex comes into being. And so we've talked a little bit about how general transcription factors play a role and help to get everything started. They bind to the promoter of genes that are transcribed by RNA polymerase II, and they position RNA polymerase II at the start site and assist in this initiation of transcription. And so usually they have these names like TF2D, TF2B, TF2A, which are transcription factor 2A, etc. And so how does the mechanism of this work? So the pre-initiation complex, this is created when general transcription factors and polymerase II we'll call it POL2, bind sequentially to the tatabox of DNA. So they will bind, we have TF2A and TF2B along with TF2D, which is this complex here, that bind to the tatabox. TF2H helicase then comes in, and it will use ATP hydrolysis to provide energy for unwinding DNA at the transcription start site and pushing that DNA through and into the polymerase. The PIC TF2D TBP subunit, which is this whole subunit here. This holds DNA into position by binding to the tatabox. So it is really kind of this portion of the complex. PIC stands for pre-initiation complex, by the way. And so this strain on the DNA, you can see that the DNA is kinked at this point. This assists the TF2B N-terminal region, so that transcription factor 2B N-terminal region, and polymerase II to melt the DNA or pull it apart. That will allow polymerase to transcribe one of the two strands. And so polymerase then initiates transcription in the resulting open complex where the DNA has been melted, and it continues transcription away or downstream from the promoter. The TF2H kinase domain phosphorylates the polymerase II carboxyl tail domain, and then the general transcription factors will dissociate and fall off, and then all we have is the polymerase group. And as you can see, the phosphorylated tails up here, the polymerase will then continue down the DNA and transcribe mRNA. Taking a closer look at RNA polymerase in the pre-initiation complex, we can kind of see with this cryo-electron microscope structure how all these transcription factors and the polymerase and everything comes together. So this is an example or a model, that's the word I'm looking for, a model of yeast polymerase II pre-initiation complex. And so this is comprised of 33 polypeptides, which is about the size of a ribosomal subunit. So it's a pretty big, hefty macromolecular macrostructure of proteins. And it has some things I want to point out here. This general transcription factor 2H, and within that, or associated with that, is this SSL2 subunit, and these work together to help open the DNA strands at the start of transcription, because remember, you're only using one of the two strands to transcribe into a gene, and the other strand is just the complement. And so what this SSL2 subunit does is it pushes DNA that is bound upstream to this TBP, which is right here, the TF2B, which is red's probably not a good color because that's the color it's indicated, and then TF2A. So what it does is it kind of threads this so it goes down in that direction. And when it does this, this creates torsion stress on the double helix, and that contributes to that transcription bubble and the melting of the DNA, pulling apart that DNA, so that the polymerase can get to and transcribe one of those two strands of DNA. How do we know what these elements are in the promoter or control region that lead to changes in expression, or vital conserved elements for gene expression? So one experiment that you can do is called a linker scanning mutation experiment. And the way this works is we take a vector, so we kind of create our own gene under a promoter we want to study. In this case, we have a marker gene, this TK gene, and what we're going to do is we have our control vector here. So we have our normal promoter and our normal TK gene, and we see and we can measure the amount of expression of this TK gene under normal conditions. And so we have 3 pluses. That's our quantifying number in this case. Now, if we want to figure out what regions are responsible for controlling this expression in that control region or that promoter, we can do random insertions of mutations. So these boxes that have these diagonal lines in it are areas where we've inserted and kind of garbled up the sequence of that control region. So it's just nonsense. The length is the same because we don't want to change the distance the gene is from any sort of upstream element, whether it's an enhancer or something like that. So the length is relatively close, if not exactly the same, but we've just changed the sequence, the order and the bases there. And so we do this over and over and over, as you can see here, a total of 9 times in this example, but probably many more than that. And we do that for the entire length of the control region. And for each of those different mutants, we measure the expression of that gene. So as you can see here, in mutants number 1, mutant number 4, mutants 6 and 7, and mutant number 9, we have no change in expression because they all are plus, plus, plus. That means the highest level expression. So changing those sequences, or the base pairs of those sequences, has no effect on gene expression. However, in mutants number 2, mutant number 3, and mutant number 5, we have greatly reduced expression. So we've turned down how many of those mRNA transcripts are made, but we haven't shut it off. And then lastly, we have an example of mutant number 8 here, where we have completely stopped transcription of that gene. So using this information, we can map those positions. So if we were to bring this kind of straight down, we can see that this conserved element here, which I'm going to try to trace as best I can, that was present in mutants 2 and 3, as well as this region that was in mutant number 5. And then lastly, this region right here of that control region, which was in mutant number 8, all have some sort of responsibility in transcription. And so what we can later deduce from this is that the furthest upstream is the promoter proximal element, PE2, and then PE1, and then the one that completely shuts off transcription is actually the Tata box. So the general organization of control elements that regulate gene expression in eukaryotes are different between Tata box genes or CpG island genes. So we're going to talk about the differences here. So most eukaryotic genes are regulated by multiple transcription control elements. That's this list right here. And so in the Tata box containing gene that we have, this is regulated by promoter proximal elements and enhancers. And these are the things that we talked about previously in the last slide we identified. And these are regions of transcription binding sites and things of that nature. So these promoter elements also position the RNA polymerase to initiate transcription at the start site of the gene, which is this +1 is where the first nucleotide is transcribed. And these promoter elements also influence the rate of transcription. So in terms of the enhancers, these green little blocks right up here, they may be either upstream or downstream actually. So in this case, you can see there's one that's way downstream as well. Or they can be within introns. So you can see here, there is an enhancer within the intron. So they can be almost anywhere except for the exon or the promoter. And they can be as far away as a few hundred kilobases from the transcription start site. And these promoter proximal elements that we talked about, they're found upstream and downstream of the transcription start site. So you can see there's a couple of them that are downstream. So similar to enhancers, they don't always have to be upstream of the start site. And they are in equal frequency before and after that start site in mammalian genes. So these CPG island promoters are kind of weird compared to the traditional gene expression and genetics. So the CPG islands, transcription is initiated at several sites in both the sense and anti-sense direction. So that's why we have these arrows going in different directions here. So sense would be the direction that is going to make the gene that we are actually going to turn into a product. Anti-sense is the opposite of that. And so the sense direction transcripts are elongated and processed into mRNAs through RNA splicing. So that would be the product here, which would go on to be an mRNA and then go on to become a protein at some point. And genes express mRNAs with alternative 5' exons, which is determined by the transcription start site. So that's why we have all these different arrows, because the start site can vary with these CPG islands on each within the gene. So one gene can have multiple start sites, hence all these different arrows. So that 5' exon, which is the dark blue here, that can be different depending on where transcription started. And then promoters contain promoter proximal control elements as well, just like the Tatabox example here. But one thing that's different between CPG, well, there's a lot of things that are different, but another thing that's different between CPG island promoters and the Tatabox is that there appears not to be enhancers, or at least isn't clear currently whether or not enhancers play a role in expression of these genes. So you see there's no enhancers upstream within the promoter, or sorry, within an intron, or downstream. They just don't exist down there. And so lastly, we'll talk about something a lot more basic. Yeast, they contain only one upstream activating sequence. We call it a UAS. And a Tatabox, which is this -90. Also, I want to point out, I haven't mentioned this, the numbers here, the -200, the -30, -90, that's the number of nucleotides they are upstream of the start site. And so that, I want to alleviate any confusion there. So they have a Tatabox that's about -90, so it's about 90 base pairs upstream of the transcription start site. So let's talk about activators here briefly. Activators are transcription factors that are responsible for increasing the rate of transcription, and they have a couple different domains. So they have two types of domains. They have a DNA binding domain, and they have an activation domain. So the activation domain interacts with other proteins, and the DNA binding domain interacts with DNA. And they always have one DNA binding domain, but they can have multiple activation domains, and they're modular as well. And so there's been some interesting experiments where you can take the activation domain from one activator, combine it with a DNA binding domain of another activator, and still have activation. And if you're interested in those studies, go look at my my genetics lecture on transcription, because I believe it's in there. But yeah, so they have these two different domains. They can have multiple activation domains, but one binding domain. Now, opposite of an activator is a repressor. So this is the structure of a repressor here. As I said, functionally, they're converse of activators. They slow down or stop expression of a gene, and they contain a DNA binding domain again, as well as a repression domain instead of an activator domain. And these proteins use that DNA binding sequence to bind to specific sequences, and this binding commonly involves non-covalent interactions between the atoms in a DNA binding domain alpha helices and the edges of the bases within the major groove of the DNA. What does that mean? Okay, so we have these non-covalent interactions between atoms of alpha helices. So let me get my arrow here. These alpha helices will form non-covalent interactions with the major grooves of the DNA. So those major grooves are the larger grooves here. So you can see we have a minor groove here, and then a major groove here. So those alpha helices kind of wedge in there, and you can see there's another example of this up at the top right there. And they form those non-covalent interactions with the DNA at those locations. So the use of these alpha helices to interact with DNA at the major groove is somewhat common. So here we have 4 different examples. We have A here is a C2H2, so C2H2, zinc finger, I'll just put a ZF, zinc finger domain. And so this is where those alpha helices linked to zinc molecules that are the dark here will interact with the major grooves of protein. In B here, we have a C4 zinc finger protein, so it's just a different orientation of the zinc fingers. You can see these are kind of angled away from you, but those are also alpha helices with the zinc molecules, and they are interacting with the major groove there. We have C here, which is a leucine zipper protein. I'm not going to spell that out. You're not going to be able to read it anyway. But we have these long alpha helices here that are interacting with the major groove. And then lastly, we have BHLH. I will write that down for you. BHLH. And this, these proteins are DNA binding helices at the N-terminal of the monomers and are separated by non-helical loops from these zinc finger or these leucine zipper regions. So really the only difference you can see here is here we have 2 very long alpha helices, and instead these BHLH are 2 pairs of these same shorter helices that are linked together. But they still interact the exact same way as the leucine helices, but they're just a little bit different conformation. In some instances, we need 2 transcription factors that are unrelated to bind to neighboring sites in what we call a composite control element. And so combinational transcription regulation is what this is known as. And this involves the interactions of these 2 unrelated transcription factors. In our example here is NFAT and AP1. And NFAT is a monomeric transcription factor, so it's just one single protein. And AP1 is heterodimeric, so it means it's a dimer. It has 2 subunits, and hetero meaning that they are different. So you can see one's blue and one's green, so they're not 2 copies of the exact same subunit. And each alone has a low affinity for their respective binding sites. And so as you can see here, weak NFAT and weak AP1 binding if they are by themselves. But when there is an interaction between the 2, and they are allowed to bind cooperatively to the composite site, which would be the DNA. They didn't have it labeled here, but this DNA, oh no, it is there. Sorry, it's just kind of covered up. Yeah, that DNA, when they bind cooperatively to it, it increases the overall stability of this NFAT AP1 DNA complex. So another example here is this SRF and SAP1 cooperative binding. And so this one's kind of interesting because SRF is also is heterodimeric. So you see there's an A and a B subunit. And then SAP1 is monomeric. But SAP1's got this interesting conformation where it also has a DNA binding domain that goes to the opposite side of the DNA. That's quite a ways, a few base pairs up or downstream. And then this is kind of a linker region that links the interacting part, the part that interacts with SRF A and B, to that opposite side DNA binding domain. So it doesn't have to be two proteins that are right next to each other. They do have to be kind of connected and present for one another, but they can have kind of extended reaches as well, and as in our second example here. So the proteins or transcription factors that bind to those enhancers upstream of the gene, it's actually usually complexes of transcription factors that bind. So this is an example of an enhantiosome that we would is the amalgamation of multiple transcription factors that bind to an enhancer. And this is for the beta interferon enhancer. And as you can see, there are quite a few different subunits that the DNA is kind of hidden underneath all these transcription factors here. But the binding sites, you can see your recognition sequence for these enhancer elements are very close to one another. And so they are kind of packed in here to aid in enhancing the transcription. And so these proteins can help produce loops in the DNA between their binding sites and to bring the DNA into the proper transcription factors and the pre-initiation complex and everything in close proximity to initiate transcription of a gene. So we've talked briefly about deacetylation and acetylation, and that is the modifications of adding acetyl groups to histone tails to allow modifications to chromatin. So we're going to look at a couple of the models of how this mechanism works and how it affects gene expression. So repressors can direct this deacetylation at specific genes in this UME6 protein here, repressor specifically. Directs deacetylation of these histone tails. And so DNA binding domain, which is this DBD right there, will recognize and interact with a specific upstream regulatory sequence, or URS1, for the gene that it regulates. So it'll be specific to a gene and it will interact with that upstream regulatory sequence. And then the repressor domain on the opposite side of that repressor, that will bind to sign 3, which is part of this larger RPD3L macroprotein. And this RPD3 is a histone deacetylase, so it gets rid of the acetyl groups on the N-terminal tails, or the histone tails, of the nucleosomes in the region where it bound. So in this like kind of local region right here, it will remove any acetyl groups. As you can see, there's no black hexagons attached to them. And what that does is that doesn't allow that DNA or those that chromatin to slide and grant access to this TATA box. You can see the TATA box is, let me get a different color, is bound up in the middle here, and there's no way that that pre-initiation complex can really get there, and so gene expression is repressed. So now in terms of activator-directed histone modifications, in this case hyperacetylation, what happens is we have GCN4, which is an activator, and it will bind to an upstream activator sequence, which is this blue box right here. And the activation domain will bind to this SAGA complex, which is very similar to the RPD3L complex, but it's an activator complex as opposed to a hyperacetylation complex as opposed to a deacetylation complex. And when it does that, the GCN5 subunit of this complex will then hyperacetylate histone and terminal tails on the nucleosomes nearby, so very similar to the repressor-directed histone deacetylation. And it helps to decondense the chromosome to facilitate access to that TATA box. So it'll allow sliding of those that DNA along those histones. It doesn't completely remove those histones, it just slides them away so it can get access. So it's kind of like a tape measure or something where you're kind of pulling it out and allowing access to that DNA. And then from there, it will allow the gene to be transcribed. And so as you can see here, the histone tails compared to the top one, they have these acetyl groups added, which allows it to free up and slide and increase transcription. So let's talk a little bit here about the structure of the mediator complex in both yeast and in humans. So the mediator complex forms a molecular bridge between activators that are bound to their DNA binding site and to RNA polymerase II. The activation domain of mediator interactions stimulate assembly of the pre-initiation complex for RNA polymerase onto the promoter. And so in A here, you can see all the subunits that make up this mediator complex. So there is a lot of small proteins that make up this large complex. And as you can see in humans, there's even more, specifically in the head group, of these mediator subunits that make up these complexes. And if you are at Louisiana Tech, most of you will be at Louisiana Tech if you are taking this class, Dr. Jamie Newman actually works and studies how this mediator complex works. So here we see the mediator complex interacting with activators and bound to the polymerase. And so as you can see, we have this is like a long loop of DNA that goes somewhere else. So these activators here bound to the enhancers are very far upstream of the gene. And as you can see, what these activators do with the mediator complex is loop this DNA around and have it so that upstream enhancer sequence is now close to the actual promoter or the transcription start site. And then we also have those promoter proximal activators that are binding and interacting with the mediator complex. And so these multiple activators influence transcription from a single promoter by interacting with this mediator complex simultaneously or in very rapid succession. And so as you can see, this kind of helps to change the conformation of the DNA and get it in proper orientation to allow for that polymerase to transcribe that gene. And so here we also have, you can see the role of the generalized transcription factors in this complex. So how is transcription regulated by kind of the extracellular environment, right? So there's different ways, but one way we're going to talk about here is hormones that bind to nuclear receptors. And so this nuclear signal, extracellular nuclear signal in this case, regulates these nuclear receptors. And so they're generally regulated by these lipid-soluble hormones, which include steroids, retinoids, and thyroid hormones. And they diffuse through the plasma and nuclear membranes, and they interact directly with transcription factors, including this nuclear receptor superfamily. So we have some example here of retinoic acid, cortisol, thyroxine, and so these are ligand receptor complexes that function as transcription activators. So here we have the general design of transcription factors in that nuclear receptor superfamily. And as you can see, they all have the same kind of motifs where they have a ligand binding domain, and that's 15 to 57% of the total length of this protein. And as you can see, they also have a DNA binding domain, as well as this variable region that is they kind of function as activation domains in most nuclear receptors. And so you can also see here that the different hormones that are able to activate transcription, such as estrogen, progesterone, retinoic acid, as we talked about. And so there's a lot of these hormones that your body obviously will put through your circulatory system, and it will go to distant parts of your body and are able to change transcription at very distant parts of your body. So here we have a consensus sequence of the DNA response elements that bind these nuclear receptors. And so as you can see here, there's kind of these conserved motifs or sequences that occur. And so in the first example, we have, or first two examples here, we have glucocorticoid and estrogen receptors. So GRE and ERE are those two. And they have these kind of anti-parallel or opposite repeats that are inverted. So as you can see here, AGGTCA, and then if you go on the opposite strand, the opposite direction, AGGTCA, and then this spacer of 3 nucleotides in between. And so this is one common motif in that regard. And then for the next, the vitamin D response, which is this vitamin D response element, you see repeats on the same strand, so AGGTCA, spacer of 3, AGGTCA. And the same thing can be said for thyroid response element. And then we have retinoic acid response element that also has that. The only difference between these three is the spacer elements, right? So we have spacer of 3, spacer of 4, and the spacer of 5. And so what that looks like when you actually put this into a secondary structure is you can see that we have this kind of symmetrical or mirrored shape for the glucocorticoid and estrogen response elements, where it's just kind of flipped, and they both interact with this major groove. And then in the case of the last three, we have these alpha helices here that are interacting with the major groove, but instead of flipped, it's just repeated, right? Over and over and over again, if need be. But for those 2 major grooves, at least in this case. Okay, as alluded to throughout the lecture and throughout some previous lectures, the histone modifications with acetylation or methylation can also lead to repression or activation of genes. And this is called epigenetic inheritance or epigenetic modification. And these acetylations or methylations can be passed down from one cell to another through during the replication process. And so in addition, these, as we mentioned earlier, these acetylation events always happen at lysines on these histone tails. And so as you can see here, K9, K14, K18, that's the number at which the lysine appears in that histone tail, so its position, and it has an activating effect. Phosphorylation can also lead to activation on these histone tails, as well as methylation of arginines. But depending on the methylation site of lysine, it can either activate or repress. And we also talked briefly about ubiquitination. And if you ubiquitinate in a lysine, it can either activate or repress depending on the position.
So how are these methylation marks maintained following chromosome replication? So this is the mechanism here. So we have chromosomal DNA replication. So as you can see, each histone here has a methyl group added, just as an example, a single methyl group. And then during replication, those histones randomly associate with one of the two daughter chromosomes. So as you can see here, we have replication and we have two new chromosomes. And at each histone, the parent one was randomly decided to be either on one or two, or the other of the daughter chromosomes, right? So half have it and half, or the other half doesn't, right? They don't both have the same histones modified because newly created histones on the daughter strand don't have those methylation marks yet. And so then this H3K9 and HMT, which is histone methyltransferase, they come in and they read the methyl groups and see where there's not a methyl group, and they will specifically add it. So as you can see here, we have a methyl group, and it will read if the next histone over has that methyl group, and it will modify and add that methylation mark to the nucleosome. And then what we end up with is a full complement of these methylation marks on the histone groups. And so they will maintain this throughout repeated cell divisions unless they are specifically removed with a histone demethylase. So lastly, we're going to talk about polymerase 1 and polymerase 3. We're going to start here with the polymerase 1. And so this is responsible for transcription of those precursors, the 18S, 5.8S, and 28S rRNAs that are used in the ribosome. It's highly regulated transcription with polymerase 1. And it correlates with cell growth and division because you need to have those ribosomes going if you're going to grow and divide. And so here at the top we have an electron micrograph, which is showing the RNA protein complexes that are transcribed from one copy of these repeated rRNA genes. So you can see a lot of these complexes being generated. And in the middle we have this single polymerase 1 transcriptional unit. We have these blue enhancer regions up here that is where the enhancers or the activators bind to. And we also have these termination sites here in red, and that is bound by pol 1 specific termination factors, which we call TTF1. And so that stops or ends the transcription. And we have these kind of exons here, these gene units, the 18S RNA, the 5.8S RNA, and the 28S RNA that are used or transcribed through this process. And then the other stuff in the middle, in this black block here, are cut out and not used. So those are kind of like intronic regions. And then at the bottom here, we have our kind of core promoter element where polymerase 1 binds to, and it's showing some of its generalized transcription factors. We're not going to get too much into detail, but you have UBF1, SL1, and TIF1A, which are these general transcription factors, and there's some other proteins in here that are required for elongation and control with polymerase 1. So lastly, we'll talk here about RNA polymerase 3. We covered 1 and 2 now, and now we're on to the last one. So this polymerase helps to transcribe tRNA as well as 5S rRNA genes. So we have, in this example here, tRNA and 5S rRNA. And these genes contain internal promoter elements, which are the yellow here. So we have A, B, and C. When polymerase 3 binds, these specific general transcription factors, TF3A, TF3B, and TF3C, will bind with polymerase to control transcription. And as you can see, in the case of tRNA, there is no TF3A, and in the case of the 5S rRNA, there is TF3A. So it depends on the gene that's being transcribed. And so the arrows on here, there's these green arrows, which indicate, let me turn this green so that makes more sense. All right, so we have these green arrows, and this indicates strong sequence-specific protein DNA interactions. So you can see that TF3C and TF3A both have strong binding interactions with DNA. And then we have these blue arrows, and the blue arrows are interactions between the general transcription factors. So you can see TF3B and TF3C, as well as TF3B and C for the 5SR and rRNA gene. Both have strong binding in between them. And then lastly, we have these purple arrows, which are the interactions between polymerase and those general transcription factors. And so down at the bottom here, we have another example. So we also have small non-coding RNAs, and these are functional RNAs that have some sort of role within the cell, and we're learning a lot about them now. They're it's a very hot area of research, and we're learning that RNAs do a lot more than we thought. They're not just messages. Anyways, back on topic here. So in this case, the U6 sRNA gene is controlled by this TATA box, and upstream of that is this PSE regulatory element, and during this transcription, SNAP-C binds to that PSE and this transcription 3B-like protein, so it's a little bit evolved from a transcription factor. It's similar, but not enough to call it transcription factor 3B. It binds to the TATA box. So you can see in this case we have two interactions with DNA, that upstream regulatory element and the TATA box. And then interactions between these three complexes leads to transcription of the small non-coding RNA, which are very short sequences. So that'll do it for this chapter. It was a long one. Tried to stick to an hour. I'm about 10 minutes over. So I will see you in chapter 10.